In exercise 1, you're going to do a size comparison of microbes. You're going to conceptualize the size of microorganisms, comparing it to a known object. Each student will be assigned a different object with a different length or diameter, and you're going to figure out how many microorganisms will fit end to end, either across the diameter of that object or along the length of that object. You're also going to review the metric system of measurement, which is what we use in science, specifically those measurements that are used in microbiology. And finally, you're going to be classifying the microorganisms in this assignment. This image shows you the relative sizes of bacteria and viruses, and it's nice to show you how much smaller bacteria are than human cells, and actually how much smaller viruses are than bacterial cells. On the right, in green, you can see a human liver cell, which is about 30 micrometers in diameter, which is 30,000 nanometers in diameter. If you imagine that green to be a big circle, you can see how much larger that liver cell is than the bacterial cell, Escherichia coli, which is down at the bottom in blue. Escherichia coli is 3 micrometers long by about 1 micrometer in diameter when it's at full size. If we convert that into nanometers, we have 3,000 nanometers by 1,000 nanometers. So it's significantly smaller, 10 times smaller, than a human liver cell. Looking above at the different artist renditions of viruses, you can see that they are much smaller than human cells and even smaller than bacterial cells. For example, bacteriophage T4 is 225 nanometers in length. As you can see, it's much smaller than Escherichia coli, so it's easy to understand why bacteria can be infected by viruses. Viruses can infect any cells that are out there because they are significantly smaller than any cell. You also want to note on this slide the measurements that we use in microbiology. You see nanometers and you also see micrometers. Typically, we describe bacteria in micrometers and viruses in nanometers, but you can see that these units are going to be interchangeable. First, reviewing the metric system. The metric system is beautiful because all of the units are in units of 10, and so you see the prefix that's put before each basic unit of measurement in the metric system. There are 3 units of measurement in the metric system: the unit of mass, which is going to be grams, the units of volume, which will be liters, and then the units of length, which is what we'll be using in this lab, which is meters. The prefix is going to be put in front of each unit, and that's going to symbolize the size of the unit. The 4 that are circled in red are the 4 that are most important to us in microbiology. The centimeter is cm. There are 100 centimeters in a meter. Just like there are 100 cents in a dollar, it's easy to remember that centimeter means 100. Millimeter is abbreviated with m, so mm would be a millimeter. There are 1,000 millimeters in a meter. Now you might think milli means million. It does not. Milli means 1,000. Micrometer is symbolized with the Greek letter mu, so mu-m is a micrometer. Micro means million, so there are 1,000,000 micrometers in 1 meter. And finally, nanometers. Nanometers we use for viruses and for widths of cell membranes, for example. Nano is abbreviated with the letter N. There are 1 billion nanometers in 1 meter. So 1 nanometer is 1/1,000,000th of a meter. Imagine that if you had a meter stick and you had a virus that was 1 nanometer, 1 billion of them would fit across that meter stick. On the upper right, you see centimeters versus millimeters. If you take a look at a metric unit, you are going to see centimeters and millimeters, and you note very simply there that there are 10 millimeters in every centimeter. There also would be then 100 centimeters in 1 meter. Again, repeating that micrometer is what we use to measure bacteria typically, and there are 1,000,000 micrometers in 1 meter, whereas nanometers have 1 billion in 1 meter. You'll see there is a difference of 3 decimal places, or 3 zeros. Between millimeter and micrometer, there's also a difference of 3 zeros. So using this, all we have to do is move our decimal point to the right or to the left to figure out the conversion from centimeters to millimeters to micrometers to nanometers. So in your assignment, you have been assigned an object that has a given length or diameter, and you're going to determine how many of the organisms, viruses or spores, on the list will fit end to end along the length or diameter of the object. The first organism is Streptococcus. It's spherical in shape at 1 micrometer in diameter. Escherichia coli, that you just saw on the previous slide, is 3 micrometers in length. Amoeba proteus is an amoeboid organism, so it doesn't have a specific shape to it. So for the purposes of this lab, we're going to say that it is 100 micrometers in length. Saccharomyces is also spherical. It's 15 micrometers in diameter. Rhinovirus, a spherical virus, is 30 nanometers in diameter. The Penicillium spore is also spherical, and it is 3,000 nanometers in diameter. And Epulopiscium fischelsoni is a rod-shaped organism, and it is 1/2 of a milliliter in length. So here's what you're going to do. First thing, convert everything into the same units. So look at the object that you've been given and convert it either into millimeters, micrometers, or nanometers. And if it's in nanometers already, you can keep it in nanometers. You just have to remember that all of the organisms or cells you are going to have to convert into nanometers as well. So everything is going to have to be in the same units before you get started with doing the math. Here's an example. The diameter of the head of a pin is 1 millimeter. 1 millimeter is equal to 1,000 micrometers. Here's that 1,000 again, so we use the multiplier of 3 zeros. 1 millimeter is 1,000 micrometers. There are 1 billion nanometers in a meter, so there are 1 million nanometers in a meter. In a millimeter. 1 millimeter is equal to 1 million nanometers. Okay. So let's look at HIV. HIV is 100 nanometers. 100 nanometers is the same as 1/10 of a micrometer. Here again, you're moving your decimal place, but you are moving it to the left. Remember, a nanometer is smaller than a micrometer. To get micrometers into millimeters, again, you will move your decimal point 3 points to the left. So you see now we have the same units for the head of the pin and the size of the virus. You can use any of these units in order to do the math, but let's use 1 millimeter. So the head of the pin is 1 millimeter, and HIV is 1/10,000th of a millimeter, or 0.0001. Dividing 1 millimeter, the diameter of the head of the pin, by 0.0001 millimeters, which is the diameter of HIV, doing the math, you get 10,000. So 10,000 virus particles would fit across the head of a pin. As long as you have the same units for the object and the organism, virus or spore, you can do the math by dividing the object size by the organism size. Remember, you want to keep these in whole numbers. So if you have anything left over, a fraction of a number left over, ignore it. Do not round up. I repeat, do not round up. The second part of this lab, you're going to classify these organisms. You're going to classify them as a bacterium. Bacterium is singular, bacteria is plural, fungus, virus, or protist. So Streptococcus, Escherichia coli, you're going to use your textbook or internet resources and determine if the organisms are bacteria, viruses, fungi, or protists. Good luck on this assignment. If you have any questions, let me know via email or talk to me in person.